everyone. Welcome to our single-use webinar. Thank you so much for joining us. My name is Marina. I'm the Digital Marketing Manager at DRM, and I'll be moderating this webinar today. It's a pleasure to introduce to you some of our newest single-use technologies, addressing the need for process identification in the production of biopharmaceuticals. So a very quick intro to our webinar and how it's going to look. So we're going to start with a really quick introduction into DRM, and then the webinar is divided into 3 sessions with a Q&A session after each presentation. The first will be about our single-use filter, the CONTIBAC, and its functioning principles. Then the next presentation will take a look at deep dive filtration data, and our last presentation will be about our single-use mixer, the FUNDAMIX SU, functioning principles and data. So let me go ahead and introduce to you our first speaker, Ivan Abramovich. He's head of our single-use business unit. Hi, Ivan. And
hi,
he
Marina.
will start the part.
All right. Thanks a lot, Marina, for the introduction. And hello, everyone, and welcome. I'm Ivan Abramovich, and it's my pleasure to have you here today. Thank you for making the time. So, in the first part, we're going to have a look at the filter, which I will present to you in a few moments. But just in the beginning, the overarching themes in this webinar are mainly scalability and process intensification, which are right now key concerns because we're facing a pandemic. And the demands for vaccines and pharmaceuticals are soaring. The first product, the CONTIBAC SU filter, is one of these solutions with which we want to help you meet those demands. And it's my pleasure to introduce you to this revolutionary technology. But before we start, I would like to give you a quick introduction to DRM, our company. So DRM, which stands for Dr. Müller AG, was founded in 1982. And ever since, we have been experts in solid and liquid separation, but also mixing. We have more than 3,500 systems deployed in various industries worldwide, mainly in the chemical field, in the polymers and oil and gas field, but also in the biotech and pharmaceutical field. We have subsidiaries in Germany, Poland, USA, China, India, and Malaysia, and the United Arab Emirates even. And we have several production facilities at these locations. But now, when you have a look at the products that are shown up here, top right, you will notice that they are all made of stainless steel, which has been our bread and butter since 1982. But as we all know, the biotech and pharmaceutical field has moved ever more towards employing single-use technologies. So this is why in 2017, the single-use project was launched at DRM, which is now the single-use business unit. Without further ado, let's have a look at the CONTIBAC SU, which is designed for intensifying your process of cell harvesting by the means of multi-cycle cake filtration. Before we start with the functioning principle, let's just have a quick recap on the status quo in cell harvesting. So what you see here on the left side is a part of your processing chain, and we are right here between the bioreactor and the 0.2 micron sterile filter. We're going to have a look at these two, the combination of a centrifuge and single-use depth filters. According to surveys, more than 80% of all processors use depth filters. So this is really what we're going to focus on today. Now, depth filters. We all know what's good about them. Let's have a look at their disadvantages, though, and what really gave us the incentive to research further into this topic. So, scaling, as mentioned. How is scaling achieved with depth filters? In this case, it's very simple. Scaling is simply achieved by stacking more on top of each other, more filter cassettes. If you look at, for example, this bioreactor, and let's say you have a 100-liter bioreactor, but you want to move to a 1,500-liter one. Well, for this step right here, this means that you have to have more filter cartridges in operation. which means that you really have to do something which is space and time consuming, and the production area is really not cheap and often very limited. And you also have a vast plastic waste when you run your process like that, which is not a good thing if you want to meet the sustainable development goals, which are also becoming more and more important nowadays. On another note, from a more processing point of view, let's say, depth filters are known to be susceptible to clogging, especially today when the industry is moving to larger batches, but not just that, denser batches, most of all, so that you go up to more dense solutions with a higher cell density. This tends to pose a big problem to depth filters. And when you're running into those risks, you have to consider that frequent replacement might be necessary in order for you to operate. And whenever an operator has to enter your production area and replace these elements or do some manipulations on the equipment, you have to validate all of those steps, and you run a risk of contaminating your production area, which is a liability. And then on another note, comparably, and my colleague Tizian will give you a very in-depth look at this fact, but with depth filters, you're working with comparably much lower flow rates, which necessitates much higher surfaces that are present in your production area. And with these higher surfaces, we come to another key problem in single-use processing, which are leachables and extractables that we want to avoid in order to provide good quality medication. But there's good news. These two steps in the processing chain can be replaced by the CONTIBAC SU. And this might look a bit, let's say, curious to you because it looks nothing like the depth filter. This is also because it works on fairly different principles. But nonetheless, you're able to avoid all these problems and really subsume these 2 points into one compact product. Now let's have a look at the functioning principle to see how we achieve that. For this, we're going to have a look at the automated filtration cycle. So here we see a schematic of the filter. What you can see here is a single-use bag, which is enclosing 4 filter elements. This is made of plastic. Now, this is furthermore closed by a pressure vessel, of which you can see the wall is right here. And in the first step, we start filling the single-use bag with cell suspension mixed together with filtrate, diatomaceous earth in most cases. So you fill up the bag more and more, and at some point, the solids are held back, but the liquid needs to escape somewhere. And it does that through the middle port here, which is the filtrate port, while the solid particles are accumulated on the outside of your filter elements. So you keep on doing this, filtering this, until you reach the maximum cake thickness. And when you reach that point, well, that would be the point where with depth filters, you would have to throw them away and replace them with fresh depth filters. But for the Counterback SU, we tend to do things a bit differently. In the second step, we commence with the heel volume filtration. What does that mean? Well, in here, you still have a lot of valuable heel volume with a lot of product, which you don't want to lose. And this is where the flexible property of our single-use bag comes into play. By combining this with a pressure vessel, we conduct compressed air between the pressure vessel and the single-use bag, really squeezing it dry of any remaining heel liquid, which is then further taken here through the filtrate port. And then in this state, we have really the dry cake accumulated on the outside of the filter elements. Now, in the next step, this is where we regenerate the filter media. In this step, we conduct a washing liquid. It can be water for injection, but it can also be a buffer. This is up to you. We conduct this through the middle port and wash down the solid remainder here, the cake. And the cake then falls down and accumulates on the bottom. The next step, we start with the discharge. For this, we open the discharge valve shown here at the bottom, and we let the liquid escape through the discharge port. And in this state, the filter is ready for the next cycle, and you can repeat these cycles until your batch is finished. So this is really how much you get out of only one bag per batch, just to put this in perspective. So we have seen the schematics, but now I also want to show you a video of this. With this, we're going to have a look at our fully automated Counterback skid. What you can see here is the pressure vessel containing the single-use filter, and you can see how it is filling up with the murky cell suspension. On the top right, you see the clear filtrate coming out of the filter. These are air bubbles right now, while the murky cell suspension keeps flowing in. And then at this point here, we have built the maximum cake thickness. And now it's time to start with the heel volume filtration. And for this, we will conduct compressed air here from the bottom, which will contract the bag more and more. You can see this is a pretty quick step. And you can see the filter elements with a brownish cake accumulated on its surface. This is what we're going to wash down now with the back flushing step. And you see how this is now filled with the washing liquid. And in the next step, the slurry is discharged, and you can see the elements are nice and white, and they're ready for the next cycle. So now you have seen how it works in real life, but let's also have a look at installation, which is also an important point, because this has to be as easy and as comfortable for the operator as possible. For this, let's have a look at the next video. So in this video, you can see the operator inserting the single-use bag containing the filter elements. You can see the transparent pressure vessel is now in a horizontal position. You can also see the pre-attached manifolds here, the pieces of tubing. This is how we deliver the product. And the operator is now closing the lid to really seal it and to make this pressure vessel pressurizable. On the bottom left, you might have noticed we have little schematics and depictions of the individual steps. Those are the pictures that appear on the touch screen that is mounted on the control panel, really guiding the operator through each and every step so no mistakes are made. In the next step, the filtration skid or the pressure vessel is rotated into a horizontal position. And here you can see the operator mounting the first piece of manifold. So that was a pump head for the Levitronics pump that you saw right here. And on this set, we have the inlet ports. So here you have the water for injection tube, and this here is the tube for the buffer. And on the very bottom, that tube is used for the cell suspension. Now the operator is taking the second piece of equipment, and he is connecting the manifold with the pump head, with the single-use pump head here. This is done with tri-clamps, but it can also be done with sterile connectors. And now we switch to the other side of our filtration skid, and there you can see the outlets. The uppermost tube is just a connecting tube. The second one is for the filtrate. The third one is for the pre-filtrate. And now the bottom one, that one is for the slurry that is then discharged after each back flushing. You might have noticed that the valves, they appear to flash in a red color when the operator removes the safety cover. And this is because they're blocked from operating. This is a safety feature, so the operator doesn't pinch his finger in there. In here, the operator is connecting the last piece of tubing. And now, as a filtration unit, the assembly is finished. The only steps left at that point are connecting the tubes to the bioreactor and eventually the place where you want to store the slurry or where you want to dispose of it. So now you have seen the installation. Let's look at how this affects your process in, let's say, the grand scheme of things. Let's take following example. We have a 100-liter batch with a cell density of 25 million cells per milliliter, and you want to scale this up to a volume of 1,500 liters. Now, what does this look like with a process that is done with depth filters? Well, it looks like this. In the first case, you might have or might not have a centrifuge in combination with this, or you might have a two-stage depth filter process. But in any case, for scaling up, first of all, you need a larger stainless steel frame, but you need several of those probably. And so you're not really working with the same setup. You have a different space, you have different space requirements, perhaps different tubing, you have different frames in operation. And really the only choice you're left with is just stacking more of these filter cassettes on top of each other, or putting them next to each other. Now, what does this process look like if you operate with a CONTIBAC SU? It looks as follows. The CONTIBAC SU can cover the whole range from this 100-liter batch up to 2,000 liters even. And it is the same filter, it is the same product, the same setup. The only difference being that you merely have to increase the number of cycles or the duration of operation. In the case of the 100-liter batch, you operate the filter for 8 minutes by doing perhaps one cycle. And for the 1,500-liter batch here, you repeat the cycle 15 times, which might take you almost 2 hours. So the breakthrough with this technology is that it allows you not to play around too much with your hardware, which is expensive and time consuming, but allows you to merely play around with your process parameters. And this is how you can scale. Now, what does this look like from a compliance and quality point of view? I also want to make a few comments on availability of this filtration skid beforehand. So, of this fully automated skid, well, what is there to say? Its commercial launch is scheduled in fall of this year. And this unit here contains the control panel and the pinch valves. And it also features this rotatable frame for the ergonomic bag insertion. It is CE certified, and its software is compliant with Hydrolex Volume 4 as well as the Code of Federal Regulations 2111 for our American customers. And from the point of view of the wetware, which is the filter bag and the manifolds, they comply with ISO 10993 for biocompatibility and 11137, which concerns sterility. They're delivered gamma sterilized. Furthermore, all the material in contact with the product is of course USP Class 6 certified, and we do an endotoxin test for each batch that we deliver according to USP 85. And we also comply with BSE TSE. Furthermore, what I would like to mention is that we produce these filter bags ourselves here in Manedorf at the beautiful Lake Zurich in an ISO 7 GMP Class C clean room. Okay. So now, what if you want to go ahead and start doing trials right away? Well, this is what we can offer you. We have, for small scale tests, the Counterback 0.5 liter. We also have the 2-liter version, and there's also a simplified 30-liter version. They differ mainly, of course, in their filtration area, ranging from 0.06 square meters up to almost 1 square meter. And of course, the batch size they can handle. This one can handle about 5 liters, while this one goes up to 50, and it goes all the way up to 2,000. The two on the left here, we offer for small scale trials. There are manual and automated versions available. And this one here we offer for pilot trials. And it is worthwhile to mention that the bag installed in there is identical as in the automated filtration skid. And that concludes the first part of this presentation. And now I'm happy to answer all your questions that have arisen by now.
Hi, Ivan. Thank you so much for your presentation. Yes, there are a few questions that came in. So first one, how long does each bag last?
Okay, that's a good question. That depends really on the parameters that you're working with. Tizian is going to show you afterwards really a very in-depth look at how many cycles we can handle. But at cell densities of 90 million cells per milliliter, we were able to finish them in 68 cycles. So this really depends on your cell density. In any case, we are able to replace really like 20-fold or 30-fold the amount of depth filters that you would need for the same process. And so what we tend to say is that each bag lasts as long as your batch lasts, because then you are basically, you have to throw it away because you want to avoid cross-contamination in the case of biopharma. If you're coming from a different field where this is not such a concern, you might even reuse it for the next batch. But there we still need to find our limit because as of right now, it's very durable. Yeah.
Thank you. One more question. How can you maintain the flow during back flushing?
That's a very good question too, because during back flushing, as the one who asked the question has correctly observed, the filter has a short downtime. I mean, there's no liquid going through the filter in that period. So there's two possibilities how you can tackle this. First one being that you perhaps have an intermediate vessel with fluctuating liquid levels, or that you have two of those in parallel compensating each other for the short amount of downtime that they might have.
Thank you so much. Do you have a comparison of yield loss volume during the depth filter charges compared to multi-cycle?
Okay, yield loss during multi-cycle. So what I think this might be going towards is that at some point you suspect that you might have some volume left in there, which would be your product, or which you then discharge. For this, we don't have a detailed analysis at the moment, but the heal volume step that we provide is really like, it looks like when you vacuum your clothes when you go on vacation. Like, it's really tight and really dry, but this is something we can look more into in the future. Yeah.
So we'll answer any other questions later on by email. We need to move on to the second session. So let me introduce you to Tizian Bucher. He's head of our R&D department, and he will start the second session.
Thank you very much, Marina. So Ivan has talked about the general functionality of our filter. I'm going to dive into how does it perform, especially in comparison with depth filters. So I'm going to introduce to you three sets of data and some general comments first. All of the cells that were used for this data was provided to us by the ZHW school in Wädenswil. We intensively collaborate with Dieter Eibel and his group. Another general comment is that all of this data was obtained at a constant pressure of 1.5 bar. And in each of these graphs, I always show the flow rate in liters per square meter per hour on the y-axis and the time on the x-axis. You can see there's two plots. The black plot here shows the flow rate over 6 cycles. The flow rate always starts high because initially you have no cake, no resistance to the flow. And as the filtration proceeds, you start building up a thicker and thicker cake. There's more and more resistance to the flow, and hence the flow rate drops until we reach the point where we start back flushing and regenerating the filter. And once that is completed, the flow rate starts high again, and so on and so forth. In this plot, you also see an orange curve, which is the average flow rate, taking into account the downtime as well. Now, this data was obtained for a total cell density of 47.5 million cells per milliliter. Let me show you the second set of data, in which we performed 20 cycles at almost 54 million cells per milliliter. And now the star of the exhibit is this one, where we did 68 cycles at 90 million cells per milliliter. Now, all the cards are on the table. Let's analyze this data. There are three things I would like you to take away from this. The first observation is 47.5, 54, and 90 million cells per milliliter. What? Can DEP filters handle such cell concentrations? Absolutely not. Second observation about the average flow rates. You can see here we achieved 445 LMH, 480 LMH, and 263 LMH. Anybody who knows anything about cell harvest knows that these are absolutely insanely high given these cell concentrations. And the third observation, it pertains to fouling. In filtration, we call fouling the successive blockage of the filter with solid particles. If we experience fouling, you would see a drop in the flow rate from the first to the second cycle to the third, and so on and so forth. But as you can see, that doesn't happen. The flow rate stayed the same over all 6 of these cycles, over 20 cycles here, and it stayed almost perfectly the same over 68 cycles here. Even though I tested here perfusion batches, which are somewhat nasty to test because there's a lot of impurities. The cells have been in these perfusion batches for a very long time. We also had no fouling, even though I tested here 2 different strands of CHO cells, and despite the very high cell density as well. So this shows that we cracked the jackpot. We found the filter medium with which we can filter at a high flow rate, a high quality, but not experience fouling. And that's very crucial, as you'll see later on. Now, you may see something interesting at this point in our filter, but there's still a chance you may forget about it in 5 minutes. I would like you to see the revolutionary in this data, and I would like you to remember this for the next couple of decades. I'm going to achieve that by comparing us to depth filters. In fact, to compare us with depth filters, I can't even use any of this wonderful data because depth filters can't handle it. So I have to simplify the problem and use a lower cell density around 25 million cells per milliliter, at which we average around 1,030 LMH. So let me put that into our comparative graph here on the right. Now, to give you a comparison for depth filters, I have a study here from 2017 in which they compared a range of different depth filters and sort of figured out the best 3 in the first step, and then tested them once more with a cell density of 24 million cells. And they reached an average of about 450 LMH. So let me put this into the graph on the right. Now, 1,030 LMH is actually not the fastest that we can do. Our filter also comes with what I call the sport mode, which is if you regulate the pH. If you reduce the pH, a lot of the impurities precipitate out, and they kind of form little agglomerates, and that makes it much easier to filter them out. And by doing that, you can really gain on speed. For example, here for 11 million cells, we reached almost 1,500 LMH. Just to give you a comparison, for a depth filter for a similar cell concentration, I have another study from 2017 with 14 million cells. They used 3 modules, all 1.6 square meters in cross-sectional area, and they averaged 500 LMH. Now, ladies and gentlemen, I would like you to appreciate one thing. Research and development nowadays is highly competitive. There are thousands and thousands of researchers striving for improvement every second, even as we speak. So progress nowadays is highly incremental. So if in this day and age, you take a competitive field such as the cell harvest, and you can say of yourself that you improve the state-of-the-art technology by even 30 or 40%, it's a big deal. It's an accomplishment. Now we come along, and I'm telling you, we improve the state-of-the-art technology not by 30, not by 40, but by 125 to 200%. That is absolutely mind-boggling. Just think about what this could mean for your process. In the past, you maybe wanted to scale up your process from 500 liters to 2,000 liters. You couldn't because you didn't want to get an army of depth filters. Now you can. or you wanted to double or triple your cell concentration, you couldn't because depth filters couldn't handle it. Now you can. You're used to thinking that the cell harvest is the bottleneck in the production of biologics, that you have to scale down your upstream processes to accommodate the cell harvest, but now you have to change the way you think about the cell harvest. There's no more bottleneck. You're free to do whatever you want. And that's so incredibly powerful. It can very quickly save you hundreds of thousands, if not millions of dollars. Now the question is, does our speed come at the cost of quality? And it actually doesn't. Here in the next slide, you can see some turbidities that we achieved for different cell batches that I've introduced so far. At 11.1 million cells, we had 4.5 NTU we measured. At 47.5 million, we were around 7 NTU. And this one was a little bit of a tougher cookie at 54 million because it had a lot of solid content. And here we were at about 22 NTU. So let me show you how depth filters compare with that. It's again that study from 2017. Again, those different models of depth filters. And you can see they also range between 4 and about 24 NTU. So it's essentially exactly the same range, and they tested that at 28 million cells. So despite having the same range of turbidity, we are 2 to 3 times faster and still get the same range. That's incredible. Now let me briefly show you some other quality measurements which we tested for this cell density, for this cell suspension here. For each of these graphs, the dashed line shows the initial value before filtration, and these bars show the values after filtration. And we did this for several different percentages of filtrate. One thing that we have studied is the activity of LDH. The LDH is an enzyme in the cells that is released when cells are fractured. So this is a way to tell you how rough a process is on the cells. And you can see here for these different types of percentages of filter aid and also for the different pHs, the increase in LDH was relatively moderate. It's less than a factor of 2, which compared to competing technologies is actually pretty good. One other thing we have studied is the product recovery. As you can see, if you do not modulate the pH, regardless of the amount of filter aid, you have 100% product recovery. If you reduce the pH, that's one drawback of pH reduction. You do lose some of the product because some of the product precipitates out as well. This here is a little bit of a worst case scenario because we used a very low viability. From my experiments, it has shown that if you increase the viability, I've tested this, for example, for 93% viability, the loss drops down to 9%. I'm quite confident that you can bring this down to, say, 3 to 5% if your cell suspension is extremely fresh. Another thing that we have investigated is the DNA, which is another impurity that you ideally filter out. And as you can see, that's a big advantage of the pH modulation. If you reduce the pH to almost 5, you take out almost all the DNA there is. Whereas if you leave the pH the same, no DNA is filtered out. And lastly, the HCP, host cell proteins, is another impurity that needs to be filtered out later on. And you can see that if you increase the amount of filtrate you use, you can filter up to about 50% out, but not more. And actually, that's one of the things we'll investigate next, is if we can use adsorptive agents, add them to the cell suspension, that then binds the DNA and the HCP and help filtering it out. It's the same thing that depth filters do. They have it oftentimes built into the depth filter medium, but we would just add it into the cell suspension, but has pretty much the same effect. Now I want to summarize this. I have shown you facts, real scientific data proving unquestionably that we are 2 to 3 times faster than depth filters while yielding the same turbidities. We can achieve 100% product recovery, and also we can handle very high cell concentrations, which depth filters can't. That means we pretty much get rid of the bottleneck that you used to have in cell harvest. And lastly, the footprint is much lower of our filter, which means less leachables, extractables, but it's also much better for the environment. So I told you, you wouldn't forget this webinar, because this marks the beginning of a new chapter in cell harvest. It's the chapter of the cyclical cake filter. The sooner you convert, the better it is for you, for your wallet, for all those millions and millions of patients out there who will have access to more and cheaper medication. It is better for the environment. It is better for almost everybody in this world. So with this overarching statement, I'm going to hand it back to Marina, and I'm ready for some questions.
Hi, thank you, Tizian. Yes, there are some questions that came in. So first question is there a mechanism to connect this technology to a perfusion bioreactor or only to a feed batch cell culture?
So the question is if this is a substitute, for example, for a Repligen filter that is used for perfusion. So this is more targeted towards fed batch, or just the clarification of the entire perfusion batch once the perfusion experiment is over. It is not intended to be used over, say, a week or two as a continuous filter.
All right, next question is, where do you get the diaphragmaceous earth? Does DRM provide it along with the filtration system?
Yeah, so we obviously don't produce the diatomaceous earth. There's very few producers in this world. We can provide it, and there's multiple providers, so it's a very easy thing to acquire.
There have been some questions regarding the filtrate. So first one was, do depth filters use filtrate as well?
So most depth filters have diatomaceous earth built into the filter membrane, and a lot of them use it supplementary as well. Because even if you have diatomaceous earth built into the filter membrane and you don't use any DE otherwise, your filter will block very quickly. So what a lot of filters, even depth filters, do is they add diatomaceous earth to the suspension and filter it along.
And one question was, how do you add the filtrate into the system?
There's various ways you can do that. The easiest way is to just dump it into your bioreactor right before you filter. That is not always possible because some bioreactors are not really made for stirring diatomaceous earth. What you could do is you could add the diatomaceous earth in two additions. Say you put some in, it tends to sediment, you filter some, and then you add some more. Another option is to have an intermediate mixing tank where you mix the suspension with the filtrate. And the third option is to have a concentrated DE slurry, which you feed into the suspension, into the feed. So you essentially have the feed, the cell suspension come from one side, and the concentrated slurry from the other side. And you kind of dosage it to the suspension.
And one other question that just came in, how to compare the filter with MF or UF?
So it's very different. Microfiltration, ultrafiltration, they use membranes. Membranes are very fine, they block very quickly. Our filter medium is much coarser than what you would use in MF and UF, and that's a huge advantage that we have. That is the reason why we do not have any blockage, whereas with, for example, a TFF filter that uses an MF membrane, you would very quickly have fouling. So the answer is it is much coarser. It is where the porosity in the microfilters are in the submicron scale. We are talking about the micron scale in our several microns.
Thank you so much, Tizian. And so let's move on to our last presentation, which will be about our single-use mixer. So for that, I'd like to introduce you again to Ivan, who will be taking over.
Hello. All right, thanks for the reintroduction. Now let's move on with our mixing products. The next product I'm going to show you is the FUNDAMIX single use, and it is a product specifically designed to provide a really vigorous mixing, but at the same time being very gentle on the cells for you to ensure to have high yields, and all the while avoiding impellers that are mostly found in other products that are intended for mixing. So on the right side of this slide, you can see a schematic of the system, or at least of its most important parts. As for the filter, we also have a single-use bag here in which the product is contained. And then in the middle, what you can see here is a center tube. It is a flexible tube with a mixing plate at its end. Now, what you can see here is a metal shaft that is screwed into this mixing plate, and this transmits the vertical up and down motion provided by our magnetic drive. But you might ask yourself, well, this is just going up and down, like how do we generate the flow in this setup? Well, this is achieved as follows. What you can see right next to it are two types of mixing plates. For exhibit A here, mixing plate type A, you can see the conical bores right here. So this plate has conical bores, and through the Bernoulli effect, we create a pressure differential across this axis right here. So this is how we generate flow inside of this cavity. We can also do the bores in the other direction, so we provide a downward flow, and in the other case, it is upward. What we are also capable to do is supply gas through the middle. But what are we able to achieve with this? Tietze will present you the exact data later on and how this really translates to mixing performance, but at the same time shear forces. But with this, by really avoiding impellers and the tip speeds that come with it, you're able to have very low shear forces, which is perfect for buffer formulation, or if you want to handle lentiviruses, very susceptible to shear forces, and cell cultures, of course. Then another thing which I want to point out here is that this system is very low maintenance. Now, this magnetic drive here, for those who are familiar with our mixing products in the stainless steel department, this is basically the same drive unit, and also it features a very similar metallic shaft. And we have units just like this one that have been in operation for more than 20 years without having to be maintained even once or having to be repaired. They're very robust because there are no rotating parts. It's just an up and down motion provided by magnets. Speaking of magnets, you might be familiar with magnetic parts in your throwaway single-use bags, which really proves to be not so nice to dispose of, but also on the other hand, a bit more expensive. In this bag, there are no magnetic components present, for example, in order to agitate the internal impeller. Another point that has to be mentioned is that there is no formation of a central vortex, which is very proprietary to impellers, which might be very damaging to your cells. With this mode of mixing, we're able to really disperse this motion through the whole batch, which can be shown later on with CFD simulations. But so like this, no baffles are required. And on the other hand, as I already mentioned before, we can of course supply this with gas, O2, or anything else that is up to your choice. And all this, of course, for you to have higher yields and have an excellent cell viability while really being able to mix this and really get your gas supply and your nutrients to the cells that you want to grow. Now, this was just a schematic. Let's have a look at a video again. Now, this here is the lab version with a stainless steel mixing plate, but the principle is the same. You can see these very visible coarse particles that are moving up and down. And you see the movement of the plate is barely visible, but the mixing is still very vigorous. That's what's so good about it. From an installation point of view, something that you might have asked yourself, how does this look like? How does this work? Well, here, just a quick example for the 10-liter version, we can see the operator handling the bag here with its key features. You can see the center tube right here, the ports on top. and which I will talk about later on. In any case, here you can see the mixing plate with the conical bearings and the discharge port. And in the next step, the operator is going to insert the metal shaft. It is analogous for the larger systems. So you see the metal shaft really has no contact with the product that is inside. And the operator is now screwing it into the plate, and this is all held together like that. And then of course we can fully customize it. This is just for demonstration purposes, just a small vent filter. But we're very flexible in any case with how you want to have it customized. But more about this on the next slide. So some key information that I would like to share about availability and customization. The compliance and the certificates are exactly the same as for the CONTIBAC SU. The materials are also fairly similar. And it is also produced in our clean room facility here in Männedorf. The ports are fully customizable, and the manifolds, of course, also, which really make them a perfect fit for buffer formulation or also for cultivating cells. You can also install a QC set. We're really very flexible there. But this port you will have always for sure, that is the one for the vibratory shaft insertion. You can have one for venting. You can also you will also have a port for liquid addition and discharge, or for recirculation, or even for solid addition. And this is available in sizes ranging from 1 liter up to 500 liters. The only limits here are, of course, is the mixing plate. And this is something that I want to stress, that I really want to stress now, because we have spoken a bit about supply shortages and supply issues. Right now, as many of you might be aware, molded tubes, TPE tubings, for example, or even single-use bags are in very high demand. There is a lot of supply shortages, and there are even some products, especially when it comes to mixing bags, that have been discontinued recently. What I want to mention at this point is that our design is very adaptable. We just need the dimensions, and with this we can work on a bag volume, and we can really fit this on any existing vessel that you have in your facility or that is on the market. And we would then mount the drive unit with an extra suspension mechanism like this, so we can really fit it into your old existing vessel, while you might then still profit off all of these advantages that we provide. And that also concludes this part of my presentation. I'm handing over to my colleague Tizian.
Thank you, Ivan. There's a few things I'd like to point out to you before we finish up here. First of all, 2 studies have been performed comparing our FUNDAMIX with rotating impellers. The first study was led by the ETH Zurich, and they compared a 4-blade impeller that you see here on the top right with 3 of our mixing plates of different sizes. And the study was twofold. In the first half, they performed a computational fluid dynamics study, CFD study, and they determined the shear stresses for all of these agitators. The second part of their study was to determine the mixing time with some experiments. And they actually did that in quite an ingenious way. They mixed 2 things, reagents that would react together, and they would measure how long it takes for this reaction to complete, and from this deduce what the mixing time is. And the data is compiled in this graph in the center. They essentially show that for the same shear rate, the FUNDAMIX can achieve a much lower mixing time if you use a sufficiently large plate compared to the impeller. Or for the same shear rate, we can obtain much lower mixing times. I mean, for the same shear rate, lower mixing times. That means in both regards, shear stress or mixing time, the FUNDAMIX excels compared to rotating impellers. The second study was performed by the Fraunhofer Institute in Germany. They took a vessel with a 100-liter capacity similar to the one shown here, and they put a bunch of conductivity probes in circles around the vessel. They put several layers of circles, and essentially what they did was they added a 50 milliliter tracer of sodium chloride on the bottom of the tank and started mixing. And with these conductivity probes and using the electrical resistance tomography algorithm, they were able to trace the path of the tracer throughout the tank and measure the time it takes until all of it sort of diffuses and averages out the conductivity. And from that, they could determine what the mixing time is. And they compared two rotating impellers. You can see here a 2-blade impeller, a 3-blade impeller, which are really typical, with our FUNDAMIX 2 mixing plate with a diameter of 135 millimeters. And this is the data they came up with, they measured. So they saw that for an A and type B orientation, the FUNDAMIX was much faster than the rotating impellers, both for water, for 50 CP glycerin, as well as 100 CP glycerin. And the advantage of the FUNDAMIX is more pronounced for low viscosities. If you go to high viscosities approaching 500 CP, this evens out and the FUNDAMIX is not as efficient anymore. Fraunhofer Institute also determined how much power is consumed by the FUNDAMIX and rotating impellers, and they found out that the FUNDAMIX actually consumes less than half of the power. That mostly has to do with the fact in the FUNDAMIX, the electromagnetic actuator only actuates one part of the motion of the plate. The other part of the motion is reverted through mechanical springs. So the system itself kind of stores part of the energy that makes it much more energy efficient. That comes in useful if you run the mixer 24/7. So far I've shown you data, shown you how the FUNDAMIX compares with rotating impellers when it comes to mixing times, power consumption, and shear stresses. Another thing I would like to touch upon is scaling, because scaling is very important in biotech. You usually start on a small scale and you start scaling up until you're at the production level. I'd like to point out two advantages that the FUNDAMIX has when it comes to scaling. The first is its uniform distribution of streamlines and its axis symmetry. As you can see in this CFD model, I start mixing here and you can see the streamlines. Well, first of all, you can see it's perfectly symmetric throughout the entire vessel. The other thing you can see is very quickly the streamlines start filling out the entire vessel. That means that the symmetry and this perfect picture of the streamlines means that you can easily scale it if you increase the vessel size, as long as you make sure that your mixing plate is scaled sort of proportionally. Then you have the same mixing performance, the same streamline picture. The other advantage I would like to point out is that if you have rotating impellers and you try to scale them, it's very difficult, because if you go to bigger vessels, you have to scale up the blades. That means you have a different RPM, you have a different tip speed, you often have a different geometry. It's a very complicated issue. For the FUNDAMIX, actually the frequency for all the models up to 2000 liters are constant. It's always 100 hertz. The amplitude is constant. It's always 4 millimeters or less. So all you have to worry about are the conical holes. And I've done a CFD study, again, of just focusing on one conical hole and changing the bore diameter. That is the smallest diameter of the conical hole. That is where the speed is the highest and where the shear stresses are the highest. And I investigated the maximum shear stresses here. I have here normalized so you can see how it increases. Now, if you want to make sure that the FUNDAMIX is scalable, all you have to make sure is that the conical hole size of the different models is about the same. In reality, it's not exactly the same, but it's relatively close. For example, for the FUNDAMIX 1, that is has a capacity up to 10 liters, you're at 8 millimeters. For the FUNDAMIX 2, which can handle up to 200 liters, you're at 9 millimeters roughly. And at the FUNDAMIX 3, that goes up to 2000 liters, you're roughly at 11 millimeters. And the second thing we have to make sure is that the conical holes are large enough so that we're not in this part where the shear rates just go wacko. We stay more in the range where on the flat side of the curve. That also allows us some benefits when it comes to scale up. So before wrapping up, let me just briefly summarize everything from this section. I've again shown data that doesn't lie. It has shown that we mix faster than rotational impellers. The FUNDAMIX also has some advantages when it comes to scaling because of its axis symmetry and comparative simplicity. I've also shown you that it's very good for shear sensitive applications. If you mix CHO cells or hex cells or viruses, the FUNDAMIX is your preferred choice. And lastly, the FUNDAMIX is also very has a low power consumption, which is good if you run the mixer 24/7. So before I hand it back to Marina, I would like to thank you for attending this webinar, and I'm ready for some questions.
Hi, thank you, Titian, so much. Yes, so let's start with some questions. Here's Ivan as well, perfect. Have you ever performed mixing experiments with biological agents such as CHO cells?
We actually did. As part of a study we've performed with the ZHW school, we took a batch of CHO cells having roughly 20 million cells per milliliter, and we mixed it for about 30 minutes before performing our filtration. And we measured the activity of LDH before mixing and after mixing, and we hardly noticed a difference from the beginning and afterwards, which shows and it proves that mixing is very gentle on the cells.
Thank you. One more question. Why are the shear stresses dependent on the conical holes?
Mm-hmm. So the conical hole size changes the speed at which, because what a cone does, it accelerates the fluid. And if you if you have a smaller conical hole, then you accelerate it through a smaller opening. At the same time, it's a little bit like in pipe flow. You leave the fluid less room, which also means that there is less, the velocity starts decreasing because the resistance increases. And that's exactly what happens. And you do that, you have rapid changes because you move the plate up and down. That's how your shear stresses are caused and increase with the smaller hole size.
Yes, thank you. And one more, is the mixing shaft also available in plastic?
So that's a good question, because obviously you perhaps want to have everything in plastic, and this is in fact really something we have been working on. As of now, we are using metal because we're very familiar with it and because it has a certain stiffness. We are still evaluating different polymers which might transfer this vertical up and down motion the best, but this is definitely one of our ongoing research projects for the FUNDAMIX SU.
Can this be used for cell culture applications?
Yes, absolutely. So this is specifically designed for being really gentle on the cells, and you can use it for adding certain additives to the cell culture, or for just mixing the cell culture itself. And but for this would take a different kind of manifolding. This can, however, be accommodated. So yes, definitely is very good for cell culture applications.
And the next question is, would it be possible to use it at 1,000 liter working volume?
Well, I can answer this. Yes, absolutely. Our FUNDAMIX mixers that can mix all the way up to 10,000, 20,000 liters in non-SU applications. In SU applications, we're still scaling it up at the moment, but there's absolutely no reason from a practical point of view why we could not do it.
Thank you. Well, then let's wrap up. Titian, Ivan, thank you so much for presentations, and everyone else, thank you so much for joining us. So thank you very much for joining. Have a nice day.
Many thanks. Bye, everyone.